Promote ID. Hello, good evening.
Good evening, sir.
Yeah, I think there is some technical difficulty for Dr. Rajmohan to enter. Shall we wait for 2-3 minutes for Rajmohan to enter? Otherwise, we will straight away start the program. So, good evening to all. Let us start the program. Am I audible?
Yes, sir.
Yeah. Okay. We will start. I hope Professor Rajmohan will be coming soon. And this is the new norm. Sometime you can enter, sometime you may not be able to enter. That is the technology. Anyway, Honorable Pro-Vice Chancellor Dr. C.P. Vijayan, my respected colleagues in the university, eminent teachers who have joined this webinar, my dear students, ladies and gentlemen, Kerala University of Health Sciences is extremely happy and proud to host this prestigious webinar on big data analysis. We have three eminent faculty with us, Professor Mohit Mazumdar, Professor Jayaprakash Mulail, and Professor Uday Shankar Mishra. All the three are the big names in this area, and we are all here to listen to them and understand what is data, big data. And big data, we all know, is the hot topic now discussed everywhere. And as a health personnel, I'm basically a radiologist. So we know how information can be digitalized. For example, a CT scan information we get in digital format. So every information now available can be digitalized. And such digitalized information can be stored, retrieved, analyzed. And such information can go into megabyte, kilobyte, gigabyte, terabyte, zettabyte, and such big numbers. Zettabyte means 10 to the rise of 21. So 10 followed by 20 zeros. So that much amount of data we are handling. And of course, that much amount of data you cannot handle by our own brain. So we have computers, we have super computers, parallel process, and so many technical things which our experts will be telling us. And these data are now used for everything. Now it was predicted that there will be a cyclone around Kerala, and that prediction was made with the availability of data, and that was analyzed by the climate professionals. And big data is used in industry, and now the consumer industry knows what is my taste, because they analyze various bits and bits of data coming from me and analyze. So things are happening. But we are in the health sector, in healthcare industry, and we should know how these data can be used. Electronic medical records is the data which we are familiar, and just imagine the electronic medical records of every citizen in India is available with India government. Just imagine. With that, government of India can create a health policy decision. So that is the range at which these data can be made use of. You can do cost cutting. You can discover new drugs. Of course, these are all the areas our experts will be saying. I should not say all these things. And we are here to listen to them. And I once again welcome all our invited faculty speakers. And with these very few words, I declare that today's prestigious webinar on big data analysis is inaugurated. I think Professor Rajmohan has found able to come. Dr. Rajmohan, you can take over and you can make your comments.
Thank you very much, sir. Thank you. We are now getting into our scientific sessions. I once again thank Dr. Mohit for joining us. And over to Dr. Mohit. You can screen share also. Maybe after your introduction, you can screen share. Thank you very much. Thank you, Honorable VC. I hope the session will be enjoyable to all. Dr. Jayaprakash Mulil, sir, and also Dr. Mishra will be joining us. Thank you. Over to Dr. Mohit.
Thank you. Thank you, everyone, and thank you. And have a very good evening to all the students, faculty, and respected dignitaries. It's my honor to be able to present and share with the presence of eminent speakers such as Dr. Mohanan here, Professor Jayaprakash, Professor Uday Shankar. I would like to especially thank Dr. Sudhakaran and Dr. Mohanan for this webinar and for giving me this opportunity to interact with all of you today. With that, I would like to share my screen. So let me share my screen.
Yeah.
Is it visible? Okay. Okay, thank you. Okay, let's start with what we have discussed and what Dr. Mohanan has been saying about big data. And today, my topic will be about big data in biology. So I'll be going through a lot of things today in a very short time. And I'm hoping that if you have any questions, you'll be writing it down in the chat box so that I'll be able to read them and maybe able to answer them now or later. So to start with, biology as data science. We are looking at biology as data science. Why? So biology is rapidly acquiring characteristics of data science. Billions of data points on genes, proteins, and other molecules that are now compiled in large files and systematically studied, as Dr. Mohanan said. The influx of this data and associated methods and data-driven research makes this field in life sciences is as prominent as the emergence of computers in technology in tech industry. So it's pretty important. So analyzing this data will lead to more knowledge and understanding about living organisms. So in this talk today, I wanted to focus this talk on big biology data analysis and discuss how a collaborative approach has helped a big scientific community to understand, extract information from these complex biological data sets, etc. But before we start talking about today's session, I would like to introduce myself and the team behind this entire initiative. So my name is Mohit Mazumdar. My specializations are in computational biology, computational chemistry, and I'm working on biomedical data sets using bioinformatics tools. And I'm one of the global representatives of Pine Biotech. Uh, Pine Biotech is a bioinformatics company that is working towards providing cutting edge bioinformatics research and education along with several other university partners. So our mission is to simplify bioinformatics and advance research through modular and intuitive platforms that I'll be showing you in the later stage of this presentation. But as my role suggests, I'm working with students, researchers, I'm providing them research support in big data analytics and biomedical data discovery. So our goal is to develop resources and capacity and bring cutting edge research resources available to a group of students and faculty for collaborations and for doing their own research. And I've been honored to be able to work with a diverse team and a renowned experts from all around the globe, including our CEO and co-founder, Dr. Ilya Brodsky, and other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background. And one of his projects is the establishment of Tauber Bioinformatics Research Center in University of Haifa, Israel. And we will be discussing the platforms that they have developed this as a university, as a research center, which is helping scientists all around the globe. So Dr. Leonard Brodsky here, he's the director of Tauber Bioinformatics Research Center. He has numerous publications in bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches for biomedical data analysis. As a team of computer scientists, data scientists, expert bioinformaticians working in the US, working in Africa, working in India, Israel, and the UK. We have a rich team and an experienced biomedical goals to help this enable this technology, bring this technology to students and researchers working in remote areas all around the globe. So as I was telling you that along with our teams and our collaborators in the US, in Israel, and also in India, we are reaching out and building a global bioinformatics community that are connected through our platforms based on research and education. So over the past couple of years, we have been seeing participants from all around the world join our training programs, take online courses, utilize our big data analytics tools in various research projects. So in today's session, we will discuss the role of big data analytics in healthcare, and we'll be focusing on more modern healthcare challenges. So the terms such as machine learning and big data are generally overused, right? And many of us have a lot of misconceptions about the use of this kind of data, the ethics associated with it, and there are several other aspects associated with this personalized data. So, however, many methods in data analysis and especially in bioinformatics, and when you're looking at genetic data, they require in-depth understanding of various machine learning algorithms, statistics, because when we talk about data, statistics is a very important aspect. And I'm really happy that today we have a session dedicated to statistics by Dr. Uday Shankara. So it would be nice to understand that how statistics play and why statistics is being used in this kind of analysis. So before we learn about all of that and the use and the understanding of this, one thing is important to understand, that why is this big data? So how can you tell that something is a big data or something is not a big data? What are the characteristics? So big data can be described by 5 V's. So first is volume. The amount of data is huge. So I'll give you an example. The transcriptomics or RNA-seq study, which is helpful in understanding the quantification of RNA. So RNA-seq study yields information about tens and thousands of genes, millions of and then there is a technology called genomics, which gives us single nucleotide mutation variants in 3.2 billion base pairs in our human genome, right? So that's like big. It's really big. Second is variety. So this data is kind of unstructured because we're still learning, and it can be assembled in different ways. So oftentimes the same phenomenon can be described in different ways depending on the kind of machine that we are using, the preparatory techniques, handling that kind of data in kind of lab settings. So that's another V. The next is value. So information we get out of the data is dependent on many aspects, including noise, technical errors, and then completeness of information in that sample. Veracity is about for example, the data that you're getting, some part of the data has high value, like it has a lot of information, and some data has low coverage or low value. So you will find that there is differences in when you see this large data and compare it within itself. The fifth is velocity. So the velocity is another important aspect, and is the change in the data types and how there are new informations that are getting generated all the time. So in biology and in sequencing itself, there's this method that we all have heard of, is the Sanger sequencing, which was replaced by shotgun sequencing. Eventually, now we are doing single cell sequencing. So we are always learning new things and have to adopt to this idea that new samples expand our understanding. Before we had before we had the chance to study everything about the data that we already have. So these are some of the challenges, these are some of the characteristics of big data. But I wanted to today talk about genetic data and how genetic data is being used in modern biology, in healthcare, in health sciences, because it's playing quite a critical role if we talk in the terms of COVID-19 outbreak. So there's a lot of it's a great example actually, that how we have evolved using this data in such in such demanding times, in such difficult times. So let's move towards that part of my presentation where I'm talking about the genetic data. So the discovery of DNA structure had a profound ability. We were able to understand what is there in the genetic code, but it took almost 50 years from that time to get to do the sequencing of the first human genome, right? But soon after that, new technology evolved. Illumina machines came in. These are sequencing machines. They are fast. The things that we did in 15 years, they could do in 2 months. And then there was a rapid growth in this. And why there was a rapid growth? Because we realized, and the whole purpose of doing this human genome project was to able to understand. We thought that if we understand all of our genome, if we understand all of our genes, we will be able to understand all the diseases. So we thought that understanding the nucleotide and giving that understanding of, because we were working on so many single experiments, we were working on a single protein, we were working on a single gene, and then we were looking at how that affects this entire mechanism. That was really, really difficult, because a disease is not so simple. And that's what we realized after that. And this huge increase in genomic, transcriptomic, proteomic, metagenomic data that happened right after that. And now it's unbelievable if you see that what kind of the amount of the volume of information that we have. So omics technology like NGS, next generation sequencing, can be used to explore the roles, the relationships, and various types of molecules that make up the cell of an organism. So many types of omics data can be generated by using NGS, the technology that I was talking about, next generation sequencing. This data shows detailed information about many different aspects. So it can tell you a detailed information at a molecular level about the genomic variants, about the pattern of genes and isoforms, learning transcriptomics, and also epigenetics, where we are understanding the methylation and those all modifications in which are not genetic. So one of the objectives of today's talk was to correlate this entire technology to translational sciences, because everyone is interested in translational sciences. Everyone wants to work on something that can help the society. So big omics data is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, machine learning transformations, selection of treatment, and those personalized cancer therapies. So pharma is using it, and you have been heard. I think everyone have heard of the different vaccines that these pharma companies are working on. There are big pharmas, there are small companies, there are healthcare companies that are working on all kind of data, and they are participating in this interconnection and the collaboration to share data now, because these data have been collected, and earlier the concept was that to use it for your own purpose, right? But now the world has changed because of internet. One of the biggest things that is helping this entire initiative, and data sharing and collaborations. So people are now more open to collaborations. They are understanding that all the skill sets that are required to do this in translational research, translational science, require interdisciplinary sciences. It's just not that you have to be good in chemistry, you have to be good in physics, maybe you have to be good in computational stuff as well. So how can you do all of that together? So that's how these companies have now come up with collaborative models. They're collaborating with labs all over the US. So even the drug that are coming up are based on some sort of collaborations. So predicting disease outbreak, digital disease detection, risk mapping, clustering, descriptive epidemiology, and the use of this data in epidemiology, and artificial intelligence and applications. So this entire consortium of big companies, small companies, research institutes, all of them who are spending millions and billions of dollars, rupees, whatever you say, into this sort of initiatives, is because they realize that the potential that this kind of study holds. And as it was told to you before also, that data is being used in every aspect, from your Amazon shopping to your research to your healthcare. So it's being used everywhere, and it will be more and more used in the future, which is not so far from now. So to learn this in little bit in much more detail, I would want to tell you some of the new technologies that is helping us, and especially in the sense of what is going on right now. So due to the advances in metagenomic sequencing, and we want to learn about the application of big data in modern research, right? So this approach allows us to dissect microbial community structures, like whatever you call microbiota, explore species richness, and then investigate the functional roles these microbial community play in many areas related to human health and disease. So due to this advances in metagenomic sequencing and other omics technologies that we talked about, enormous databases of sequencing data or sequence data from such communities has led to the emergence of this field called computational metagenomics, which can help study 99% of uncultivated microorganisms in the context of their natural environment. So I was talking to you about how experiments have been done before and how they have evolved and changed, and why this becomes such a all of a sudden a big data, and big data in biology, big data in terms of life sciences, natural sciences. Why? So this is the reason, because the way this evolved, initially it was really challenging. Now there is a possibility to do it uncultivated. So that has changed, and that has helped us to gain all this information. So in the context of information, this information is becoming widely accessible. So it's just not that it's accessible to the companies, it's accessible to everyone, almost everyone, because you need to deposit this data to be able to publish, to be able to apply for grants. So this millions of sequences and hundreds and thousands of full genomes are available on repositories hosted by NCBI, National Center of Biotechnology Information, and other databases like Joint Genome Institute, Integrated Microbial Genome Databases. So the databases are growing in size at an exponential speed. You can see from this graph. And it's easy to access this data. It's not so difficult, because we do understand that to reach out to a large number of people who are accessing the internet, they require simplified way to understand things, because everyone is at a different stage. Some people in the US are experts, some people are India are really expert, but to be able to reach out to a lot of people who have to understand this, because it's related to their health, their healthcare, our healthcare system. The entire scientific community is developing these like a graphical way to look into the data and to be able to extract this data. So it's not so difficult anymore. So metagenomics refer to both a research technique and a research field. Metagenomics is the field that can be defined as the genomic analysis of microbial DNA from environmental communities. So metagenomics tools enable the population analysis of the unculturable or previously known microbes. The application of this technology is proving very important in some of the ongoing research. So the next part of my presentation will be about a new field of research, a very important field of research, not a new field, but a very important field of research because of what we are going through right now. So that is the application of bioinformatics in infectious disease research, and these analysis methods that are used in viral evolutionary studies, antiviral drug discovery, as well as emergence of drug resistance in bacterial pathogens. So in the last 20 years, availability of genomic data has led to an increased understanding of how cell function, so how cell function at a molecular level, and has allowed us to understand major causes of deadly diseases, right? So with this current rise in viral outbreaks and this deadliness, there is a major focus on developing tools and data repositories for better understanding of this emergence and prevention and treatment of infectious diseases. So as a result, the major advances in data generation analysis methods and utilization of insights from such studies is making a completely new approach to diagnostics, disease characterization, and also development of interventions. So vaccine development, for example. So the emergence of this genomic medicine is really helping us and to treat, to start, is helping us to ability to detect, study, and then treat infectious diseases. So these achievements came as a result of close collaboration between computer scientists, biologists, and then bioinformatics is playing a critical role in all of this. So in the past couple of months, we are working with our participants to work on some of these aspects using the platforms, which I wanted to show everyone, because many of these resources are free to use, but it's kind of useful to be able to do this, to learn, because usually that's the understanding also, because I think we believe that you learn the most when you do it yourself. So I can talk about big data, I can talk about the characteristics of big data, but unless you see the data and you run your own analysis, the challenging you will never face the challenges that are associated with big data. So there are several challenges, and these people are some of the students that are working with us and working on several bioinformatics projects, especially in COVID-19 research. Shrija from IIT, And this collaborations are intercollaborations is what something that we are promoting that helps people to collaborate and do some research that is useful and that they can present, that they can showcase and help them learn this in much more detail. So today, as this is a session that talks about the analysis, I wanted to show all the students and the faculty some of the resources that we developed for big data analysis. So one of the key resources is the Tauber Bioinformatics server built for clinician, biologists, faculty, universities to do this kind of research and establish uh, research in several healthcare domains or biotechnology, even agriculture. And this for learning coding for people who have no exposure to coding and are biologists, and this to learn bioinformatics without any experience in bioinformatics at all. So we believe in this project-based methodology because I was talking to you this that what gives us real experience is to able to work, understand the technology, and then apply the technology. And then when you apply the technology, you are working and you're doing it for a noble research. So that gives you a real experience. It's an experience that many of us have got in our PhDs, but to bring this to a level of an undergraduate level or a high school level or a postgraduate level was a challenge. So this company and our company Pine Biotech has been helping students and researchers and the university to be able to achieve that within a very short period of time, and using some of the tools that have been designed to help people. So let me just go and show quickly that how this entire thing works and integrates these all three platforms into a project-based learning. So I will just go to the browser, and this is the link for the server, and I have already logged in, and you can see that there are several sections to analyze the data. And when you hover around, you will learn about that what this technology is. There are several data types. So one of the big data type is NGS data, which we talked about. So if you're talking about RNA-seq or gene expression data, then we talked about the genomic variants and the genetic modifications, if we want to understand and learn about that kind of data. And then we learned about different type of non-coding RNAs and those kind of data. And then there is metagenomics data that we discussed in length. And then there are mass spec data, structural biology data, because once you learn about the gene, once you learn about the protein, the next step is to learn about the structure, the function of those individual mutations, how they affect the function of the protein that is leading to the disease. So getting that information from a genetic level, from a personal level, to come to the structural level, this is a complete journey. This is a complete research journey. So there are several other sections that were developed by collaborative work between UCSF, UCSF and Pine Biotech, and other collaborators from Georgetown Medical University, LBRN. So these are the universities that participated in development of this kind of analysis platform. So let me give you a quick overview and show you example of breast cancer cell lines, breast cancer samples from humans, and compare them and show them how this entire thing has been made easy so that anyone can do this analysis and like learn from it. Because it's important that biologists and clinicians and students also get an idea of what kind of genomic data that we have, how to understand the outcome of the such analysis, and also to be able to be more aware of what is going on in this modern world, where these companies, where this all healthcare system is evolving and changing. So I wanted to show you all that there's a demonstration pipeline based on a publication. So this publication was so maybe I can show you the publication if I have it here. So as I was telling you, that this is something that is developed by Tauber Bioinformatics Research Center in University of Haifa, Israel. So many of 90% of these tools are available for as online because they are published and developed by different academic groups. So many of them are open source. So 90% of them are open source. The reason to do this in this place and to create this is to learn about the logic of the analysis, that why we are doing this, why we are comparing this big data, and how what we can expect from doing this kind of study. So to be able to do that on a scale for a group of students, this is really helpful. So when I click on start, it's a point and click system, and this is a AI guided system. So it tells you about the entire data processing methods, why we are doing this, and usually we use this for teaching students and also for training purposes. So the next step, so we already have downloaded and inserted all the files here, and these files we described. So these are two groups. One is ER positive, another is triple negative. So these are two subtypes of breast cancer. So due to limited time, I will not be able to go into the details, but we are comparing two type of breast cancer to understand that if some of these genes can help us identify that kind of cancer. So I'm thinking and I'm talking in terms of a biomarker discovery. So these tools are for pre-processing. So the platform is color coded, which means that it helps us to understand or develop a logical understanding. So blue section is for pre-processing, cleaning those technical problems from the data. So we will be removing the technical sequences that came from the sequencing technique and are not biologically relevant. So this part is called pre-processing, and you can obviously read about it when you click here. It shows you the entire like tutorial about it, that why we are doing pre-processing and what is the importance of this technology. So this is meant for going through one by one step. So this is the first step where we are doing pre-processing. Then we are mapping then we are comparing this transcript to a reference, because in when if we want to understand that how I am different from another individual, or if I have a disease and the other person is a normal person, so we do a comparison, right? So this is also a comparison where we are comparing these samples with normal samples, and to see that what is the difference between a normal person and a person that have a breast that have these genes, express genes in a breast cancer condition. So as I was telling you, that it's a point and click system to learn, so that we understand the logic of the analysis, and then we can perform our own experiments when we have a research question. So this kind of study and this kind of education is leading to research based understanding, that how research is helping in understanding of these diseases and these mechanisms and henceforth. So what I did here is that I built a pipeline. So this is a pipeline that comprises of different logic, different sections I used. So the first section I used was for pre-processing. This is what I did for mapping on the transcript. So I'm mapping it into the reference genome, human reference genome, and then I'm generating a table of expression. So why I'm generating a table of expression? I'm generating a table of expression to learn about the differences between the normal genes and the highly expressed or the low expressed breast cancer genes. So if I click N, then it will finish the pipeline and I'll get results. I'm sorry, something happened. Let me try again. So usually the way it works, that it shows result that we can interpret and open in Excel and do the downstream analysis to be able to help understand that what is going on. So the next step after doing this processing is applying statistics, because when you get data, when you get from raw data to a table, so you get an Excel sheet, And with that Excel sheet, you can apply a lot of statistical methods like t-test. Then we have methods that are for bigger data sets like DE-seq2. So usually that's what this we go through in our training sessions, that how we can understand and analyze this sort of data, and then come up with research proposals. So you see here, I got all the results in text format. I can download them, and then I can learn that what I have got from this. And this entire thing is described in our courses. So we have written a lot of courses for students, like they are like tutorials based on publications. So the publications have data sets, so you learn how to interpret a publication. And that's what we are doing in collaboration with universities, as I was telling you. Going back to the presentation, so this kind of training, that this is a very unique training, and that's why I wanted to tell you all. This omics logic training is something that is being done for the first time, and it includes a collaborative approach from several universities. And I'm reaching out to universities in India to be able to adopt this. So we do sessions, and then there are these tutorials, and then they're coding, and then this platform. So this entire experience helps us to understand how a research group published some some well-digested, good publication out of this entire analysis. For example, whether they're doing on transcriptomics, genomics, metagenomics, and machine learning. So that is what we are working on, and we are collaborating with different universities and coming up with these sort of programs where we are helping in infectious disease research, transcriptomics data, machine learning, and different other applications of bioinformatics. So as a result for a faculty, it's really very helpful, because when the students are trained, they actually are participating in a group as a group research, and the faculty is able to carry on his own research with the help of this research ecosystem that is being created due to this sort of platform access to the students, and easy accessibility, and a lot of communication between the science that is going on. And a lot of students were really excited, and that's what we have found in India so far, working for a couple of years. Students from Kolkata, a lot of faculties from Kolkata, where we started this, and now reaching out to other places. And this is a special project from a student. So she's an undergraduate student called Sarah. She presented and did this project on transcriptomics analysis of tardigrades. So tardigrades have a unique stress adaptation. So looking into that kind of data. So students are coming up with really nice ideas, and they've been able to work on some of the data that is available publicly, and come up with some scientific analysis that is leading to, that is helping them develop research methodology, understanding a publication from the very beginning. And also they are able to present this work at and publish these sort of poster publications, and maybe if they work for a longer period of time, also publication. So this is something that I wanted to show everyone, that that we have been doing in collaboration with universities in India and US and Africa. And I hope that this entire presentation was useful for many of you. And with this, I would like to ask if you have any questions, and if we can, if we have any questions and any suggestions for me.
Thank you, Mohit, and thank you very much. You have simplified it to the extent of understanding by even a layman, and you have also specified all the applications. That is very interesting to us, because it shows a shift in the paradigms of epidemiology, modern epidemiology to molecular epidemiology, transgenomic epidemiology, and computational epidemiology, digital epidemiology, etc. So now we move on to kindly use the chat box, and Mohit, you can just surf the chat box and answer using the chat box. We are actually short of time. Now our great teacher, Dr. Jayaprakash Mulil, is here. So I request sir to take over as the chair, and our session will be starting with Dr. Yadav is interested in statistics and computational aspects, and Yadav will be presenting using the screen share. And another professor, a public health expert, Dr. Biju Soman, professor from Achumenon Center for Health and Social Sciences. He has a hands-on experience in using big data analysis, and actually he works mostly on descriptive epidemiology, especially spatial analysis of data. And he is also in charge of many Government of India programs related to these things, especially in surveillance of infectious diseases, modeling of infectious diseases, spatial analysis of infectious diseases, etc. So thank you, Dr. Biju, for joining us. And I hope you will also explain in a very simple manner. And over to Jayaprakash sir for controlling this. And actually our other speaker, Dr. Mishra, also is here. Thank you, Mishra sir, for joining us. Over to Jayaprakash sir.
Thank you, Raj Mohan. I hope I am audible.
Yes, sir. Yes, sir.
And I will not take up any more time because I'm also looking forward to this presentation. This is the game of the day, and we got to be familiar with it, live with it, and learn it to be with this particular time and space. So over to the speaker.
Yeah, the unmute ചെയ്യണേ. Yeah, the unmute ചെയ്യണേ.
Can you hear me?
Now we can.
Can you hear me now? Okay.
Now we
Yes.
can.
Okay. So today we'll over the next 10 minutes I'll be just rushing through the scope of big data in epidemiology. The previous speaker has spoken elaborately on the various aspects of big data. So I'll be just deviating from the slides. As you mentioned, there are various V's when it comes to big data. We'll be discussing about each one of them, how it is related to the health. The volume it can run into, as you all know, it can run into zettabytes. Here, the volume can be imagined in this way as an Excel with the data going longer and longer into millions and billions, and the data getting wider and wider in millions and millions. There is especially different from our traditional data, and this poses a lot of issues to the analysis. And when the data becomes longer and longer, that means when you just take an Excel with lots of data, there's a sample size will be more, and whatever you get, you will get p-value in every cases. So here comes the importance of knowledge in the domain. Without the normal knowledge in the domain, whatever the results you get will be significant. What is the problem with the data getting wider? That is another issue. Data getting wider means we are familiar with only some 200 or 500 variables in a database. But when it comes to the big database, the data will be enormous. That why it will be wider. Can go into thousands, 10,000s, millions. So preconceiving the hypothesis or imagining the hypothesis or bringing into your structural causal model or
concept will not work there. You need various kind of machine learning algorithms that is different from our own method of analysis. Now, when it comes to volume, how you store the data, that is also very important. We all usually store the data in premises in our own computers. But when it comes to big data, it is better you store the data in the cloud. There are a lot of platforms like Azure, Amazon, AWS, etc. Then when it comes to examples of the big data, we have genomics data, then we have the administrative data, and we have a lot of data coming from the social network and wearables. Now the new thing coming up is the wearables. It can, from moment to moment, it can what do you call it? Can accumulate the data from your body. Then another database, big data we have is the e-health data or healthcare data. Then you have the insurance data. So if we can combine all these databases with the data linkage system, that means you have an enormous database, multiple databases with the disparate and disparate origins. So if you can utilize all this data and analyze, it can result in various hypothesis generation and insights and even solutions. Now, another issue, another thing with big data is the variety of the data we have. Traditionally, we are concerned or we are accustomed only with the structured data in the form of Excel data, because everything is structured with the schema and everything. But when it comes to big data, there is unstructured data, semi-structured data like that. Semi-structured data means you have the email data or semi-structured databases data, or you have the unstructured data from the medical data, like sir mentioned. CT images. Then you have data from various other medical records. We have a lot of data lying around in various hospitals untapped, like in the form of test case sheets. A lot of data is there. Then we have the video data. So this variety of data can be combined in big data. Then another thing. Here, the problem is when you have the variety of the data, there comes the issue of validity. And the data may not be extracted or mathematically accumulated just for the particular purpose. It has been accumulated, it has been stored for some other purposes. So there will be an issue. One of the things the epidemiologists can do is they can take an active role in the designing phase itself. There comes the importance of designing the e-health systems with the semantic interoperability with our database, so that we can pre-plan what are the things we want, and we can have it in a structured way. That means we as epidemiologists we have to influence the data gathering process to improve the validity of the administrative collected data. Then, when it comes to the variety of the data, traditionally we are using Excel, or we are using RDBMS. That is means a relational database like in the form of MySQL, or legacy systems in the form of Excel and Access. But in the big data, you need to store the semi-structured data also, and unstructured data in the form of videos, audios, photos, etc. Here the problem is our normal databases will not take this. So we need some other kind of databases. Databases like SQL. Then another issue in big data is the velocity at which the data is being produced at every moment. The next moment it can become irrelevant. A lot of data gets accumulated at every moment. One of the well-known examples is the Google flu trends. After some time, the whole scenario changed. It became irrelevant. Then another most important thing is the veracity. Veracity, as the previous speaker mentioned, it is about the trustworthiness of the data. The data has to be trustworthy. It depends upon two important factors. One is where it is sourced from. Next, what is the context from it has take origin. These two things are important. Otherwise, it will result in a lot of biases. The most of the important problem here is the ratio between the signal noise ratio. If the veracity is not correct, the noise will be more than the signals. So it will result in various interpretation issues and correct result getting. Before you processing the big data, you have to go through a lot of steps like cleaning, pre-processing, etc. But there is another view. The sheer enormity of the data itself obviates the need for theory and validity. It's a controversial issue. Because the data veracity is an issue and you don't have a theory, and the data as such, it is a data driven. They say it is a data driven procedure. The data driven procedure it will see the hidden patterns the normal method cannot detect. and it will result in the new hypothesis, or it can even answer your questions. So these are the areas where we have fundamental difference with the data, but that can be actually corrected with the involvement of the epidemiologist at the design stage itself. Then the volatility of the data, that is very important. How long it is important, that is another important thing. Then the complexity. Actually, the complexity means all these factors contributes into the complexity of the data. Here comes the importance of the how you scale up. That means once you have the data, data size increases, how you going to tackle that. Here it is very important. That is different from your normal data analysis. Here the scaling up is not an issue, not a good option. Why? Because if you increase the RAM, or if you increase the GB, it doesn't solve the issues, because that cannot tackle the big data. That means you need to scale out. That means you need kind of parallel processing. That means you divide the chunk of the information you need to tackle into different systems and tackle it parallelly. That means you have different systems, and each systems you push the data. So you in the same parallel way you process the data. Here it has got flexibility also. Now, the solution, as I mentioned, traditional, there is a big difference between these. Now, the traditional data, usually we use the RDBMS, like your Excels, and big data use NoSQL. NoSQL, the most famous one is the MongoDB. Then you have other, various other database also. There are differences between this, how data is stored, etc. Then I was mentioning about the Hadoop distributed, that is a parallel computing. This is very important in big data. Big data has to be in a parallel setup. Here there is a particular setup known as a Hadoop system. This Hadoop system that can be adopted in R or Python. We have various distributions. So this Hadoop can Hadoop along with another generic function called MapReduce. MapReduce is very simple concept. That means you divide the activity into multiple chunks. Then you map into different chunks and allot it to particular server. Then you combine this. Then you accumulate. That is a MapReduce function. So we need, the most important thing, we need to have data handling framework. Usually this could be coded, or this you can use ready-made solution like the previous speaker mentioned. When it comes to epidemiology, there is a particular word called analysis gap. The big data is getting larger and larger, but the analysis we do is less and less. The insight that can be attained from this and from the analysis of the big data is actually that getting wider and wider. So here is the importance of epidemiology and the domain expert playing a big role. There is a lot of untapped potential we can have here. So actually, it is about the synergy. It is not about we learning the algorithm. It is not about we learning the mathematics. It is not we learning the statistics. It is about understanding the terminology. It is about how to talk to the other persons in the domains. We can't learn everything. This is a collaborative work. So it is about the synergy of the data scientist, statistician, and computer programmers. As we all know, as epidemiologists or clinicians, when we do our analysis at the design phase or at the analysis phase, we should have the help of that statistician, because they know better than us, though we do know how to do that. Then it is a synergy of various experts. Here the domain experts is also very important, because if we are studying something big data related to traffic accidents and the pattern and how it occurs, and then you need a domain, you need a surgeon there. You need a what do you call planner there. There are a lot of things domain experts needed. Then an epidemiologist is also needed, because he can design the study at the initial phase itself. He can design how the particular data can be taken. Then we need computer scientists also. With the linkage is very important, because the data is spread across various disparate databases. So this database has to be combined. So we need linkage systems. Then when it comes to cross-domain collaboration, one of the things that we don't understand as epidemiologists or a clinician don't understand is the terminologies. So the most important thing is the terminological acquaintance. What label is very simple. Our Y is the label. Y means outcome. Our outcome, they call it when I do a machine learning, they call it we call it labels. Features means our variables. Variables are known as features in machine learning. Labels are our outcomes. Labeled data means there is a data labeled with Y and X as present. Then the outcome dependent factors and independent variables are there. Unlabeled data means you don't have the Y, you don't have the Y, but you have all X's, you have all variables. Unlabeled data, so there's a difference between labeled data and unlabeled. Then there is a difference in the terminology when it comes to sensitivity and positive predictive value. Sensitivity is known as a positive predictive value is known as precision in their parlor. Then the difference how they use regression and classification. We know our logistic regression is actually included as a classification model in the under machine learning. And the regression actually is only for the continuous variable. So there is a difference in the terminology. Logistic regression means it's a classification algorithm for the machine learning expert. Regression means it deals with the continuous data. So then that is this kind of terminological differences we have to be very acquainted with. Then another important thing is, as I mentioned you earlier, the Excels or the database, when the number of observation is known as instances in the instance, a particular instance. Instance means an observation or a row in an Excel. This become larger and larger. It goes into billions and billions. Every result, every P becomes significant. What will be the clinical significance? And what usually we do is regression analysis in these kind of settings. But regression analysis has a problem with the scaling up. With this enormous data, there is a problem with the scaling. So you need other kind of algorithms. There comes the importance of all other machine learning algorithms. Then another issue, as I mentioned earlier, it is the wider data. When the data becomes wider, we cannot start with a preconceived conceptual framework, or we can use it. We can, if it is a causal model that we are developing, we can never start with something called structural graphical models or anything. There, because the number of variables are enormous. So you need some kind of data reduction methods. You need some kind of automatic variable selection. There are various algorithms in machine learning where machine learning experts do the automatic variable selections. So these are the issues, because we have to look from their angle, through their eyes, but always making sure that the theory is not bypassed, though the hypothesis or the new knowledge can be generated from the enormous data. Then there is any kind of statistical methods or machine learning algorithm can be applied in our big data, like artificial intelligence. Then even deep learning and supervised. There is a difference between supervised and unsupervised. Unsupervised learning means there is no that means the model is developed inside itself only. Then okay, then towards the last of the slide. So actually the frontiers are unlimited with well laid out data handling framework. That means we should have a system and a cross-domain collaboration. There is enough fun needed. Actually, the big data analysis can help us a lot. But there is a new term coming up. That means combining the traditional data into total data into combining the big data into traditional data so that we can have the total data. So in a nutshell, the most important thing is we don't have to learn all these algorithms. You don't have to go and read all this, learn all this coding. What we need to do is understanding the terminology, understanding the philosophy, how to talk with them, how to have a mindset to collaborate with them. It is a collaborative work where the various domain experts, along with the epidemiologists and the computer specialists and data scientists, come and bring their view table. Thank you.
Thank you, Dr. Yadav. That is an excellent overview. I will reserve my remarks for the time being. I request my good friend Biju to make his comments.
Thank you, sir. Professor J.P. Mulel, sir, can you hear me?
Yes, very well.
Okay. Thank you, sir. Thank you, everyone. Let me thank our honorable VC Mohanan Kundumel, sir, and Rajmohan, sir, for giving me this opportunity this wonderful seminar. I really enjoy the presentations. Dr. Mohit has given us an orientation on the technical aspects, and Dr. Yadav has given the importance of using big data analytics in healthcare. So now we have a small group of us working in Srijitra in the Achyuta Menon Center for Health Science Studies. A couple of my PhD students and me, we are interested in data science. So that's how we got into a bit of experience in big data analytics. So I won't go to the technical aspects, but some of the concerns of using big data analysis in healthcare and the potential of using that will be discussed. So when we talk about big data analytics, actually it all started with a love affair. It was back, John Bush, actually, he was teaching his girlfriend. John Bush is a trained theoretical physicist. So his girlfriend was doing PhD in biology. So when he was helping her in data analysis, then something struck him that the biological data also can be looked upon with the sort of expertise that he developed in his physics background. So there actually they have this sort of very structured biomedical sort of understanding. So data is a problem, you split that problem into a small workable entity, and then try to answer how can we answer that. So scaling down the problem into workable questions and then trying to get the answer for that, and then generalizing that inference into the wider community. So that was a sort of approach in physics which they were using. So he found that the same thing can be done in biology, although biology has very complex data structure and all. So the initial, one of the initial attempts was looking at Alzheimer's disease. Like in Alzheimer's disease, there is a lot of gender disparity. 2/3 of Alzheimer's disease occur in women. So why that is happening? And then they were, at that time, the genomic data was also coming. And when they look at, they could see that some of the genes which cause Alzheimer's disease, actually those genes are really activated. For men and women, certain women, their response to that gene, to their hormones were different. So then one hypothesis came up, whether we can use this hormonal replacement therapy to bring down the risk of Alzheimer's disease in the added risk of Alzheimer's disease in women also. So if you want to study that, usually in our conventional way of research, probably we had to go for an RCT and we had to wait for quite a long time, or we had to look at genomic development and all. But something struck him that actually, why don't we gather data from the previous records? All women who were using hormone replacement therapy, all records, if you couple together, then whether we'll be able to address that thing. So that was a wonderful initiative. And that's how till now, quite a lot of big data analysis in healthcare is on genetics. And that is, in a way, that is good also, because in genetics, we have quite a lot of database, very structured and very accurate database, and concepts of big data analysis can be applied there and all. But when it comes to other health aspects, as we all know, in epidemiology or in health, when we are looking at the determinants of health or the social determinants of health, or even the clinical determinants of health, is varied and very complex. And people, when they started applying these big data concepts into the health scenario as such, there were a lot of, in genomics, actually, there were a lot of successes and all. And when it started applying into healthcare, actually, there was a lot of expectation that it's going to solve quite a lot of things at all. But the major drawback came from that aspect also. Like Dr. Yadav was mentioning about the Google FluNet and all. So in 2013, when influenza struck the USA, the Google FluNet predictions was much higher than what the conventional methods predicted. And actually, it turns out that FluNet prediction was wrong. And that caused a lot of setback to big data analysis. People started questioning the methodology, whether, like in research, means research. No, if we use the same methodology next time, we should get the same assumption. But in big data, that is not possible because data is pouring in. So next time when you do analysis, you are getting quite a lot of different sets of inferences. So that causes setback also. Like the strength of big data is in finding out the associations, but how the associations relevance in healthcare or in the causation and all, actually big data has nothing to do that. And for that, actually we need health people. So that's why I'm requesting that more and more people from health domain should get involved with this big data initiative. It's a multidisciplinary process. Actually, this is where probably for health or public health or clinical people, we can think about this big data analysis is not a research at all. We are not getting involved with this dirty business and all. But in the world, they are going to use big data analysis. Whether we get involved or not, people are going to work and they come up with the results and all. But we getting involved with that in the process, probably we'll be able to bring in positive changes or from the very beginning, we can tell them about the importance of understanding the data and all. For example, data has a history. Data is not without history. It's not ahistorical or it's not apolitical. The very nature of data, how we collect the data, that data, if we are using for some other purposes, it will have its own problems. What we call in reproducible research, arcing, that bias kind of things can occur. And the data, who collect the data, who controls the data, whether data is shared between government and public agencies. So whenever in big data analysis, we have to work with many collaborators. We can't restrict to public or private. We have to work with public, private, and so many agencies. And whenever health data is coming in, so when we are comparing data from different sources, and sometimes the privacy issues comes. When we are pulling in data, we will not be, how can we ensure the privacy of the data, confidentiality of the data. Now, for that, we have to have a clear data policy in place. So these are some of the issues in big data analysis. Unless we have clear policy guidelines, then sometimes it could be riskier, because data or information is now wealth, and information in the wrong hands may not do good for the society. So for societal goodness, or societal advantages, taking advantage of the newer technologies, probably we have to take a proactive step. If you look at the adaptation of newer technologies, you can see that newer technologies, usually that favors the privileged and people who have extra time, resources, etc. and all. So we have to be very cautious about that and all. That is why when we are looking at the data, big data analysis, then we are using all the data, but all the data means only digital data. So digital data, we can see that there is the issue of inclusiveness. Quite a lot of data is in the paper form. Probably the most of the underprivileged sections, their data won't be in the digital form and all. So people are using Google. If you are looking at Google Trend alone, then you are telling the story of a different strata of population, not the entire population. That also we should be looking at it. So that representatives of the data is very, very important. And then another thing is that this sort of probably this big data analysis is very good in public health. For example, in public health, we have to work with varied set of data and all. And that gives us quite a lot of opportunity to collect data, and probably it will help us to understand the social determinants in a better way. But for that also, probably many often informatics people will jump into some conclusions. But probably health or public health or clinicians should be there to warn them that actually what we are seeing is just associations. And because when we are using quite a lot of data, there will be quite a lot of noise also. So there will be quite a lot potential for a lot of confounding factors. So we have to see through that confounding factors, and we should be very judicious in interpreting big data. So with that, let me stop it here, and I'm open to questions later. Thank you.
Thank you, Biju. And both of you have really laid out the background and the intricacies, anxieties, potential of big data analysis. I'm very grateful. Now, a question to Dr. Raj Mohan. Should I permit now questions, or will they use a chat box?
We usually encourage use the chat box
Thank you.
because
Then
everybody
I'll
may
make a
have
few
questions.
comments.
Kindly
of my
make
own.
your comments, sir. Thank you, sir.
1985 was a year when I met Professor Gitelson, a professor in Johns Hopkins University. He showed me an analysis he had done. He divided all the women who gave birth to the children in the previous year in United States and divided them into two groups. Those whose maiden name was the first 13 letters of the English alphabet. And the other comparison group were the remaining 13 letters of the alphabet. And he found that the mean birth weight of babies born for the two groups was statistically significant. The p-value was 0.0001. That is impressive. I think you should remember this when you name your child. Now, the problem is somebody asked a stupid question to him. Dr. Gretelson, what is actually the difference in the mean weight? He said, glad you asked me. It is 0.00001 gram. Now, big data, as Yadav and Biju pointed out, come with a lot of issues in interpretation. And the way you present it may confuse the heck out of people. I don't know whether you know the whole story of an American railroad company who started a new railroad, and to work on the railroad, they took 100 healthy young men. But to cook for them and make sure they were fed well, they also took 2 young women. At the end of the whole show, they came back and the newspaper report said 2% of the men married 100% of the women. Now, it's not only a way of your data and the way you interpret and the way you understand. But you made an important statement about information is wealth. Well, the other way to say it is you can make money out of information. The problem is that this is something that is happening. And that's why big data is popular. I will not go into detail, but Kathy O'Neill, a very competent statistician, she wrote a book. And the name of the book is Weapons of Math Destruction. Weapons of Math Destruction, Kathy O'Neill. It is unfortunate that this particular technology can also be misused. You would all seen Dr. Piquet, she called him Piquet in that Piquet, our celebrated hero in Google, who had to face the Senate interview about how personal information was being made available through Google, big data analysis. And I tell you one thing, I have a midline approach to most situations. I think big data analysis is here to stay and you should use it. But you should use it not to conclude, but to start thinking. It is a hypothesis generating process. You can make money on the side, that's your problem. But in public health, I will be cautious because we have a responsibility in public health to people because our existence is for the public and their well-being. Companies will do all kinds of things because they exist for their well-being. So with that, Dr. Rajmohan, I'm so impressed with the presentation, and I'm grateful for your invitation to be with you this evening. Good night.
Thank you very much, sir, for joining and your valuable contribution. We are all really blessed. Now we have to go to Dr. Mishra. Dr. Mishra, as you all know, is a scientist and he is a professor in statistics at Center for Development Studies. He is having a lot of PhD students, and most of his PhD persons are working in many reputed institutions. Over to Dr. Mishra. Thank you, Mishra ji, for joining with us. Dr. Mishra. Thank you.
Yes,
Thank you, sir.
Dr. Raj.
Thank you.
Just 1 sec.
Thank you very much.
Well, let me thank the Vice Chancellor of Kerala University of Health Sciences and Dr. Raj Mohan to have invited me to speak on this forum. Can you hear me?
Yes, sir. Yes, sir.
Hello?
Yes, sir. We can hear. You are audible.
Yeah. Let me begin with a caveat because I must tell it to everyone that neither I am a big data specialist nor I'm a person who actually does work on big data in health research. Having said that, I agreed to speak on this forum because I'm motivated by the fact that there is a lot bit of confusion with regard to what is big data, what is e-data. And in fact, over time, information overflow has been the real crisis for research. And offers an opportunity, but sometimes it also has the tendency to confuse people. So before I say about scope for big data analysis and its statistical issues, as Professor Jagras Mulil rightly gave some instances and example which tells you that where you should be careful, where you should be cautious. But in fact, I wanted to clarify one thing here, is that in health research in particular, there is two broad divisions, one broad divisions in health research. One is the people who work with micro data, people who work with hospital data, people who work with lab data, but there are also people who work at the level of the population. So definitely the analysis that it entails need not be kept in divided segments, but they need to be brought together. That job has actually been somewhat eased with the availability of information all around and the information overflow, or recognizing every information to be relevant for doing health research. Say, for instance, where does this big data, the concept of big data, I was listening to Mohit, I also listened to Dr. Yadav, and where does it come from? In fact, big data has a scope which is far and wide. Big data can be starting from emails to in public domains, you have transactions, you have commercial outlets, you have social media, everything generates big data. Therefore, it is called broadly and agreed upon as an unstructured data. But here I wanted to make a statement. In fact, it is named as big data, but actually for research, there is a kind of a step. One is called information. We transform information to data, and then from data to application and utilization or treatment of the data to infer something. So every information getting converted to data is itself a very big exercise. Whether that facilitates the requirement or not, does it actually come into or entail certain kind of treatment or not, is very, very important. So actually the big data coming from a wider sphere, it is very, very important to recognize that all the type of data that comes from is not necessarily or easily or readily treatable. Therefore, information to data transfer, data transition is a very, very crucial step in big data analysis. Therefore, I say that actually the big data challenges that is coming, it's growing quantity on one hand, it's quickening speed, that is at the speed it comes in, it continuously updates itself, and the type of data that is also contained in big data. There are three things. You have a large quantity, constantly increasing, constantly coming in and getting updated, and the kind of information that comes in needs to be converted because all information are not of the same type. Therefore, big data characteristics are like this. But we must agree that big data gives an opportunity of also a very rare kind, because of the fact that actually it has never been realized that big data or the information that is available at hand, it makes better informed decisions. It also discovers hidden insights. It also says, how do you automate things where actually certain things which are identified becomes useful. So in fact, these 3 points that I have written here in the slide is that what is informed decision about? Informed decision about is how much macro can you go? How much greater, wider you can speak about? Because every inference needs to be, everybody has an anxiety about an inference. How generalized can we make it? How more, replicable we can make it? So that is where big data comes into question. During my lecture, definitely I come into what is called the big data approach and not merely the big data by itself. So given this kind of a situation, look at this, and this is the central slide of my presentation. The central slide of my presentation, I say, because there is something called an analytic model. An analytic model has different levels of difficulty and different levels of valuation. And that I say is that, as I just now mentioned, you have information at the bottom, and from information, you are going to optimization. If you can see 3 words that I write, information is in the hindsight. You derive the insight, and then you make the foresight with that. So from hindsight to insight to foresight is the pathway. Actually, the data analytic models thinks of beginning is that whenever you have any information in hand, the first thing that you can tell, you can make a sense of is the state of affairs. Then your next order of anxiety goes on to saying what is its associated characteristics.
Hello.
It comes to whether those, on the basis of the second step, you will be able to do something as predictive. What will happen? Will these characteristics actually lead you to say that what will be the predictive component? And after the 3 steps, there is the final step where we talk about that we have information to optimization. When we do, on the basis of information, the translation that we makes a prescription in ultimate terms. And that's what we give name as policy, we give name as intervention, we give any name to it. So what I wanted to actually spend time on is that...
Sir, your screen is not shared, sir. Sorry to interrupt. Your screen is not shared, sir. If you have slides, sir?
Yeah, it is shared only. I saw it is. It shows it's shared. Is it not
visible? I don't know why.
Is it not
Not
showing?
seen, sir.
Is it showing?
Not showing.
Not showing now?
No, it is not.
It is not
It is
seen.
not showing, sir.
Not seen, I think.
Can you once more do it, sir?
Yeah,
Sorry.
just 1 sec, 1 sec.
Thank you.
Let us see, Dr. Rajmohan, we should share the screen. One sec, just I'll resolve this matter for you. Yeah. Is it coming now? Are you able to see?
No, sir, it has not come.
Did it come?
No. You may have to. അതെ സ്ക്രീൻ ഷെയറിംഗ് നമ്മുടെ കുഴപ്പമല്ലല്ലോ അല്ലേ?
അല്ല.
I think it is saying that somebody else's screen cannot be shared. It is something of that kind. Sharing files. The initial slide, nothing shared, nothing came in the hello.
Nothing.
Nothing came.
Because the question is that your share screen says that you share. It says one participant can share at a time.
Oh, I see.
Sir, are you using a different machine for the slides?
I am in a Mac only. No. Same machine.
Dr.
And,
Yadav, Dr. Yadav, have you stopped sharing?
Hello?
Dr. Yadav, have you stopped sharing?
Yeah, I stopped sharing.
Okay. Mr. Sir, even without the slides, your presentation was very clear. That's why we haven't disturbed you.
I can share because the problem is I have never failed in sharing, but somehow this is telling share the screen. When I say it is saying one person can share or not, something like that. And when I say share, allow Zoom to share your screen. Open system preferences.
No problem, sir. No problem. You can continue, sir. Don't bother about it.
Okay.
Okay, sir.
Okay, fine. I mean, I can send to Rajmohan the slides so that he can share with the participants.
We will upload in the site, sir. Thank you very much. Thank you.
Yes.
No. If sent to Dr. Rajmohan, Rajmohan can.
Yeah. Please continue,
Yes,
sir.
May I continue?
Yeah, please continue, sir.
Yeah. The question is that I was talking about the central slide where actually it is about how from information to optimization you move with the difficulty levels that you have a descriptive analysis, diagnostic analysis, predictive analysis.
Sir, you please share your video, sir. We want to see you.
Oh, sorry. One sec. Can you see me now? No? Okay. Is that fine?
Perfect.
Yes, sir.
Thank you, sir. Thank you. Thank you. Fine.
Okay. So given this analytical model, I go on to saying what is the data mining technique. In fact, in data mining technique, I have given some names to it as what is called data classification, data clustering, data association rule discovery, sequential pattern discovery, then regression and deviation detection. Why I say this as data mining techniques, this does not cover everything, but it does cover the preliminary component of data mining. Data mining, when you have information with you, you often try to do a classification because data cannot be read without classification to make any inferences. Why clustering is important? Because actually the first basis of data clustering analysis is about identifying geography of data, actually identifying basic characteristics, that is geography of data. The association rule discovery is the second step in my earlier slide that I was trying to show to you, which you can see that when you go from descriptive to associative characteristics, from state of affairs to the kind of what actually is the characteristic state of affairs. Now, sequential pattern discovery is also a descriptive component. Regression is a predictive component. And this particular thing which I am detailing to you is primarily linked with what I said as when you start with this step of saying that how do you go from descriptive to diagnostic to predictive and then prescriptive analysis. So given this kind of data mining techniques at hand, what we do is that now I come to the fact that what are the challenges facing biomedical big data? The reason that Dr. Jadhav rightly told, the amount of information is abysmally large, actually lack of organization of this information, as well as lack of access to this data, and more particularly, the tools to analyze such data. Then, as Mohit rightly told, they're promoting exercises, insufficient training in data science methods that handicaps use of biomedical big data. This is the challenges that you have. Now, let me deal a little bit about introduction to healthcare data analytics, because data analytics in health is slightly different from any other sphere, because data here come in many forms. Those forms determine what can or cannot be done with the data, because data offers its own limitation. You can do certain things, you cannot do certain things. For instance, we have, like in survey research, called temporal and cross-sectional information. There are techniques for temporal information. There are techniques for cross-sectional information. You can do, you may not be able to do. But big data have a potential to actually advance healthcare research in many ways. And here I come to the fact that what is more important is information to data. That exercise, big data offers different packages, like which was mentioned in Dr. Yadav's presentation, as Hadoop and MongoDB kind of tools are available, which information to data you can convert. However, biomedical big data face many other challenges, just not that the information being there and information that you want is available and you can make or maximum or optimize use of it. Having said that, analytics actually is an entire process. It is not merely what we do. The analytics, when I say it's an entire process of data collection, extraction, transformation, analysis, interpretation, and reporting. Now, when you have this whole thing constitute analytics, there are different types of data which determine what can or cannot be done, that I already said. There are various technologies or tools for working with different data types. Because, for instance, one example I can give you, which is very, very important, is that when you have a survey information in hand, you have an administrative information in hand. In fact, currently I'm engaged with an exercise which I'm actually breaking my head on, is that why this conflict between administrative or program-based statistics, which are available, vis-a-vis the statistics or the estimates that we obtain in surveys. And this disagreement actually leads to a lot of controversy in policy circles, a lot of controversy among administrators, whether they want to believe their data or they have to believe the data that surveys and other exercises bring in. The estimates do differ. Therefore, I have been given this assignment to say do's and don'ts or use and non-use of which statistics in which space and in what kind of a situation. So various challenges are faced when working with big data is because of the fact that this is another which is going beyond survey, where you can obtain certain inferences, you can obtain certain kind of conclusions, which will again come in conflict with the kind of very convenient administrative data that is available. I must tell here that actually information overflow is not an issue. Issue is about recognizing interaction between information sources. And that is what I'm coming to particularly focus on now. That is called the statistical issues. The statistical issues of big data. The first one is the ecological interface. And second thing is that the big data information will come as information and what I name as denominator-free information. You have an information, you have a particular information relating to some preferences, relating to some kind of thing. Its characterization may not be there. You might have to proxy those characterization. And there comes one lesson that we must learn, which is very important for health research. What I name is triangulation of information across sources. Why triangulation is very important? Because, for instance, you have a survey, you have a kind of a statistic which is available based on a micro data. Nobody makes an attempt to make that statistic or that particular estimation being relevant and estimated for the entire population. When I say triangulation of information, it also is important that characterization and triangulation going hand in hand can actually make your data set more relevant and more useful to infer on a larger scale. Depends on information set, because with or without characteristics quotient, if you have an information set which does not have characteristics, you perhaps cannot do, but you can definitely scale it up to a larger level. Now, one exposition that I want to do, for instance, what big data can give. I mean, this is just a quick off the hand example that came to me when I was preparing for this talk, is that consider an information of drug sell in a certain locality. That you have, the big data can give you that which regions has what kind of drugs dominantly being sold. And this kind of information is going to be available to us. Like once someone told me that in the United States of America, in the evening at 10 o'clock, they can find out that how many kilos of potatoes were sold in the whole country. Why? Because these are all digital. Nobody sells anything without entering. So the entire collation of information can give you that how many kilos of potatoes were sold. Similarly, what inferences can it offer if you have a drug sell information in a locality? Say prevalence of a certain element, concentration of specific vulnerabilities. These are inferences which are not scientific, but these are inferences which are possible to derive when you have information given by big data. Health information needs essential triangulation with population information. And that's my viewpoint, because of the fact that if health information that is collected in a very, very micro way does not matter. But thing is, at population level, there are information which proxy health information. Disability, for instance. Why we are talking of concentration, clustering of these characteristics is because of identifying actually the geographic space. Big data is misunderstood as limited to electronic records. No, actually big data, as I saw beginning told, that it has a diverse source or diverse areas from where big data originate, and it represents data other than health as well, which is often available through other sources, like for instance, consumption pattern. It is a great and potential link with health status. Ownership pattern. We feel that ownership pattern is not important. For instance, if you want to look at sedentariness of a population, can it not be derived based on ownership of personal vehicles? Because ownership pattern, consumption pattern are available, which are not to be, I'm not even stating that it has to be derived from surveys. Big data can always say which regions consumes more of certain things than other regions. which regions, which locality, because of the fact that the big data identifier always will be disaggregated in the geographical scale in the beginning. Now, overall, as I stated to you, these are very, very important, that we actually feel that big data can facilitate health research. My first point is that recognition of big data in health research and its population frontiers, because big data information need not be directly health information. Which you are talking of is collation of e-data. That is one thing, but actually a whole lot of information, behavioral, whole lot of information in relation to the consumption, the asset, the kind of spending, the kind of preferences, everything comes with big data. And if we actually become more intelligent, these all variables have a strong association with health. Proxying information that is not directly health related, but behavioral to associate with health research is a very key departure that big data can make. Triangulation, which actually I have been focusing on, remains the key. Identifying pattern and finally moving from individuals to households to communities to regions. These triangulation of information had been an exercise in a time when I grew up, when information was scarce. So triangulation had to be done. The exercise, like in statistics, we have a tool called simulation, which has perished because of the fact that simulation was actually generating information based on very limited clues at a level that was later exercise was conducted as to this is the larger information. At a time when there was no cost affordability to obtain information, as well as information was pretty limited in terms of its content and in terms of its spread. Information to data, data to receive treatment, that is called methods of exploration, and finally, inferences to qualify statistical strength. These are the three levels through which we have to go. Information which is available. In fact, the entire exercise of big data is nothing but information. Now, first task that is at hand is to information to data. How do we make? And then the data to receive treatment. The data has to receive treatment, and those are nothing but methods of exploration. You can have umpteen set of methods for exploration, but methods for exploration has to fit into the data type. Methods of exploration has to fit into what exactly the data type, characterization, as well as the domain of data content. And finally, inferences that we make have to qualify statistical strength. I emphasize on that because it is about replicability. It is about generalization. It is about concluding with greater confidence that this inference is true and is true, which can be taken into policy. These are the kind of big data application strength that we have. And in fact, whatever little that I had in my mind, I share with you with one very small quote at the end. What is statistical strength and what is? It is better to be vaguely right than being precisely wrong. This is my message when we deal with big data. It is an advantage. You can be right vaguely, but that's much better rather than being precisely wrong when you rely on all statistical strength, but not having a hold of the entirety. So the take-home message is better become vaguely right than being precisely wrong. Thank you.
Thank you very much, sir. Now, in fact, we have nearly 10 to 15 minutes. If somebody wants to raise questions, please use the chat box. Kindly look to the questions in the chat box, sir. I request Professor Sudhakaran, sir. Sudhakaran, sir, you are there? Sudhakaran, sir, PR Sudhakaran, sir. I think sir has left. Because sir is an expert on molecular genetics and bioinformatics. Sudhar sir is there? Okay, I have seen him, but now not responding. Okay, no problem. We have a little time. If anybody want to ask any questions, can ask directly also. Maybe we may go for 10 minutes or something. Dear participants, any questions? The chat box, I don't find. I have one question. A vast big data analysis has some sort of uncertainty associated with every inference. Is there any way we can quantify or adjust for that uncertainty? I think this is to basically to the statistical concern. Mishraji, can you answer to that question?
Yeah, can you repeat the question for me, Dr. Asma?
As big data analytics has some sort of uncertainty associated with every inference.
Yeah.
Is there any way we can quantify or adjust for that uncertainty? Because basically handling uncertainty.
Yeah.
Like sensitivity analysis we do for other informed decision-making support tools. Can we have something like that here?
Hello, sir. I'm Dr. Arun. It is my question, sir. If I may, can I ask you in person, sir? My question was, sir, when we talk about big data analytics, there is a lot of processes that are going in the background, and not always they are evident and very clear. And every inference that you get is packed with a lot of uncertainty. Is there any way we can quantify that uncertainty, or we can adjust for it, or is there any way we can go around it, or can yeah, that was my question, sir.
You see, I must begin by saying that any big data information that you have at hand, you do not have to accept with any kind of certainty of it. Because what happens is, first stage, as I say, from information to data, information is a clue. It's a clue. For instance, I gave you an example that, for instance, you have big data information about the number of cycles sold. This has nothing. Who bought? You don't know. There is no certainty. Okay. Now, I'm telling from a business side thing that this information is available. Should this information be not used? I will say no. It should be used, because that information has a valuable connotation towards what is the behavior of people in terms of adopting cycle right. Okay. Now, for instance, you have an information set, as you say very rightly. There can be uncertainty with regard to what information comes in and what information gets overlooked. Entirely, you will not get the information. So the problem is the big data providers, they themselves have a role. For instance, when elections are getting predicted with big data, means what? People's attitude, people's feeling and perceptions in terms of their communication are getting adjusted. How it is getting adjusted? That is getting adjusted with certain degree of uncertainty, undoubtedly. But there is a kind of a clustering. There is a kind of a counting of who are the people who react in this way on social media. Am I right? Who are the people? So the question that you are raising, despite the uncertainty of big data, or despite that, you still have a clue which you have to intelligently use to go into the next stage, or what I say is information to data. And then you can use it for your further treatment.
Yes, sir. I have a small follow-up question, sir, if I may. I was more concerned about the process. For example, in neural networks, it is not always evident on how the inference has come out, or how the result has come out. So in that case, is there any way we can quantify that uncertainty? That was my question, sir.
Perfectly fine. For instance, you see, when you do on the basis of a scale of understanding of information obtained in big data, to categorize. For instance, you want to categorize. Categorizing is a subjectivity attached there. This categorization can be on a hierarchy, best to worst. Categorization can be on a kind of a scaling. So the categorization is your choice, obviously. So what you are doing here, I'm telling the information you are using to categorize. That means you are going to data. You are getting my point. From going from information to data is your task. And in fact, there is no uncertainty there. And you can be very explicit about it, that how did I do it? How did I group it? How did I categorize it? So therefore, I told you ecological interface, when you say across regions are characterized or categorized. So categorizing it exercise itself involves two things. It's a hierarchy, that is your own hierarchy created, or in categories which are objective categories. Am I right? So this is an example I'm giving you, because uncertainties can be resolved by you. Big data will not resolve the uncertainty. Provisioning will be done by big data.
Sir, would it matter if we go for scaling or any other method? Would it?
I mean, that's a whole range of methods available. You can choose any one. That's up to you.
Okay. Thank you, sir. Thank you. Thank you very much. Sir, you are muted, sir. Rajmohan, sir, you are muted.
Rajmohan sir is muted. Yeah.
JP sir.
Dr. Rajmohan. Yeah. Dr. Rajmohan, please unmute.
I have unmuted. It is Jayaprakash sir. I think sir has left. Okay. Any other panelists to make any more comments? Mohit, you are there or you left?
Yes, sir. I am here.
Okay. Do you want to make any more comments about the uncertainty handling?
No, I think it was well covered, but like it's a broad topic.
Thank you very much.
Yeah.
Thank you very much. Okay, thank you very much. Now I invite Dr. C.P. Vijayan, who is our Vice Chancellor, to give the vote of thanks.
Respected Vice Chancellor of Kerala University of Health Sciences, Professor Mohanan, Professor Rajmohan, the respected panelists, I'm here just to express my vote of thanks. First of all, let me thank our Vice Chancellor for giving permission for the conduct of this webinar on big data analysis. Thank you, sir. Thank you, sir. Thank you very much. And Dr. Rajmohan, he needs special mention for organizing this and for piloting these discussions. Thank you, sir. Thank you very much. Our thanks are due to Professor Mohit Majumdar for giving an overall idea about what is meant by this big data and how what are the uses of big data in various fields of modern science, including health scenario. And we are also thankful to the presentations by Dr. Yadav and the discussions offered by Dr. Biju Soman of Achyuta Menon Center of Health Sciences. It was very useful. And Professor Jayaprakash Mulil, he has chaired that session, and he made it very clear how big data can be misleading in the field of public health with his very apt examples. Thank you, sir. Thank you very much. And the last session on data analysis and scope for big data analysis by Professor Udaya Shankar Mishra. Yes, it was a very, very useful and thought-provoking, and there were a lot of questions and discussions went on very well. And your final comment that it is better to be vaguely right than to be precisely wrong. Thank you, sir. Thank you very much for these comments. And once again, I'm thanking all the panelists and participants, and also everybody who made the discussions very active. Thank you. Thank you very much.
Thank you, everyone. Thank you.
Thank you. Thank you, everyone. And the contents will be uploaded in the site, university site, and the matter will be also available in the YouTube, in the site. Thank you all. Thank you very much.
Thank you. Sir, may I stop the session?